All right, so I'm Vivian. I'm a senior at Basis Independent Silicon Valley, and this is like, yeah, this is my senior project. Should I start? Or okay, so this is like about the synthesis of novel fluorinated corticosteroid analogs of dexamethasone as anti-inflammatory agents. And this is like a brief outline of everything I'll be saying. So starting with a brief introduction on corticosteroids and the specific one in this project, which is called dexamethasone, and then onto some computational modeling and docking, to find binding affinities and things like that, and then synthesis of the actual analogs, and finally assays and RT-qPCR. So this is a brief introduction about corticosteroids. So corticosteroids are a class of steroid hormones, and they're mostly man-made. So most of the really potent and strong-acting ones are man-made. Like most of these here are man-made except for hydrocortisone, and these all have the same like ABCD ring structure of a corticosteroid. And these are anti-inflammatory drugs that help against diseases dealing with inflammation, such as rheumatoid arthritis, asthma, a lot of skin rashes, and allergies, and a lot more. And these are just examples of other corticosteroids. So here is a brief like introduction to fluorinated corticosteroids. So this is cortisol, also known as hydrocortisone. This one doesn't have a fluorine at this carbon here, so all of these are C to fluorine bonds of corticosteroids, and these are a lot more stronger than things like hydrocortisone, because this carbon to fluorine bond helps with the metabolic stability in the body and prevents it from being like broken down by a lot of stressors when it is taken inside the body. And then it increases its lipophilicity, which basically means it can interact with the cell membrane easier. And corticosteroids generally work by crossing the cell membrane and then releasing like its effects. And also just generally retains molecular structure and function of the actual molecule as a whole. So this is the binding mechanism of corticosteroids and how they kind of work. So this is a corticosteroid. And because they're steroids, they kind of work the same way. So they basically bind to a receptor. And this is GR, which is the glucocorticoid receptor. And this binding event basically leads to a cascade of things, which eventually leads to an anti-inflammatory gene to be transcribed. So this gene is like acetylated, so it's able to be transcribed. And it makes mRNA that eventually helps make anti-inflammatory protein. And then this anti-inflammatory protein allows the actual person to have like anti-inflammatory effects. So this is dexamethasone. Dexamethasone is a glucocorticoid, which is a class within the corticosteroids that bind to the glucocorticoid receptor, which is also known as GR. And this is obviously an anti-inflammatory agent, and it inhibits the NF-kappa B signaling pathway specifically. And then the NF-kappa B signaling pathway is involved in a lot of immune responses inside the body. So by inhibiting that, we have less inflammation, which makes it a really good anti-inflammatory agent. And this is kind of a picture of how dexamethasone interacts with the protein. So this ketone group right here, which is just the oxygen, a hydrogen bond to a tryptophan, which is an amino acid on the protein. And these two hydroxyl groups then hydrogen bond to the asparagine, which is shown over here, like over here. And then finally, this C/C ring fluoride, which is, as I talked about before, helps improve the stability of the molecule and absorption by the body and how it works. So the goal of this project is to create dexamethasone analogs, basically. And then this is dexamethasone again, which is an FDA-approved 1958 anti-inflammatory agent. And through like the synthesis with selective alcohol functionalization, the goal is to create analogs at C11, which is this carbon right here. And this is through things like, number one, computer model informed binding thermodynamics. So lots of computer modeling and docking to kind of find out, like kind of get a sense of how it works before actually trying it out, and finding the role of the C11 stereochemistry. So this carbon right here is like, there's like this wedge here, a solid wedge here, which kind of means it's kind of pointing out at you versus like a dash here, which is like away from the screen. So finding out if that like stereochemistry really affects its capabilities of being a good drug, and finally improved the target selectivity by how like well it binds to the glucocorticoid receptor. So here are some analogs that I kind of found online. So here are like these are like just really common analogs of current corticosteroids are used a lot. So I guess, so for most of these, like the first word, like betamethasone or triamcinolone or whatever, is the actual corticosteroid. And then the word after, like valerate or like furoate or acetate, for example, is like the actual functional group attached to it, which makes it like an analog of that corticosteroid. So I kind of use really commonly like use analogs that work really well and attached it onto dexamethasone. So here are the ones I came up with. So there's an acetate group here, a furoate, and then a dipropionate group at C11. And then I made kind of like two different versions and stereoisomers of the same analog. So the difference is just in like the stereochemistry at this carbon, which is just like the solid like wedge or the dashed one. And I kind of ran it through computational modeling. So like the tan structure is just the glucocorticoid receptor, and then this molecule is dexamethasone. So they all kind of bind differently to the glucocorticoid receptor based on like the actual analog. And these are the results I got. So binding energy basically means like how well it binds to the glucocorticoid receptor. So the more negative the binding energy, the more strongly it attaches to the active site of the receptor. So my results kind of like found that the dexamethasone dipropionate, which is the most negative, kind of worked the best. But all of these work better than the actual control, which is dexamethasone without the analog. So onto like the actual synthesis of the analog. So first, so this is dexamethasone again. The importance of, I guess, protecting the hydroxyl groups here is to prevent them from reacting, because we only want to add different functional groups to C11, which is this one. So there's like we would kind of looked into two methods of doing that. So first one is like adding an acetonide group by linking these two hydroxyls together to make this. This one kind of yielded really bad results because it's harder to like close this ring, but the TBS one worked a lot better, and we're able to get a lot more products from this. And by protecting this hydroxyl group, you can kind of make different add on different esters and ketones at the C11, and then deprotect at the end to get the actual analog. And these are just different reaction mechanisms of doing that. So in order to exactly elucidate the structure of these analogs to see if we got like what we actually wanted to get, we use like nuclear magnetic resonance spectroscopy, which is just NMR spec. And how it kind of works is that because like each molecule has like atoms with nuclei, they kind of resonate at different frequencies when you put them in the NMR like instrument, which basically produces this graph showing the different like nuclei at different frequencies. And then through a bunch of like reasoning, you can kind of correspond like each atom to which peak and see exactly if you got what you wanted to get. And this is an 1H NMR, which is just looking at hydrogens. And then to kind of test the actual results of these drugs, we use inflammation assays. So first, it was important to kind of figure out, compare like the actual inflammation levels of the different corticosteroids at lab. So at lab, we had dexamethasone, which is the actual corticosteroid I was looking at. There's fludrocortisone, prednisone, triamcinolone, and mometasone, furoate, and hydrocortisone. So we kind of want to look at all of these and then the actual dexamethasone analogs. And how they basically work is that we chose the Chinese hamster ovary cells. So CHO cells are epithelial cells, so they're more likely to actually be involved in like inflammation and things like that. And then put them in like into like these wells and actually assay them. So basically, you treat them with the actual corticosteroid treatment or analog treatment or whatever, and other substrates that are inside the actual like inflammation assay kit. And how it basically works is that the activation of caspase-1, which is this enzyme, leads to a bunch of like cascade events, which activates luciferase. And the more activation of luciferase there is, or like the more caspase-1 activation there is, kind of signifies more inflammation. So luciferase, which basically produces a glow, the more glow there is, or like the more bioluminescence there is, then there's more inflammation. So more effective corticosteroids and more effective analogs would have less of the glow because there's less inflammation. And then this bioluminescence is measured with the instrument. And then finally, to identify markers of inflammation, the first step was to grow the actual cell culture and then induce inflammation with cytokines. And what cytokines kind of do is just like mediate the inflammatory response and things like that. And the ones here, IL-6, IL-10, TNF alpha, are all involved in actually inducing inflammation and adding like more inflammation. So by inducing inflammation, we can kind of see exactly which genes are involved in inflammation, like a lot. And then mRNA is extracted to get the mRNA and put into RT-qPCR. In RT-qPCR, the mRNA is then transformed or like synthesized into cDNA, complementary DNA, and it is useful and it helps identify genes of interest that are actually involved in an inflammatory response and new pathways that we can look at that dexamethasone might be able to target. And so this picture over here is kind of how it works. So this is a one-step RT-qPCR where mRNA and primers, which help actually synthesize the new DNA, and reverse transcriptase and a bunch of nucleotides are added into a mix, and then it is put into the instrument to get a graph. So this is more specifically for polymerase chain reactions, but how it works is that you first have the DNA with the sequence of interest that you want, which is denatured, and then annealed with primers. These primers just help start like it transcribe a new strand. And after many cycles, you have, after like 20 to 30 cycles, you have like millions of copies already. So the difference between polymerase chain reaction and RT-qPCR, which is reverse transcription quantitative polymerase chain reaction, is that RT-qPCR starts with an mRNA, which is more specific for actually identifying gene expression related to like inflammation. So in RT-qPCR, because you start with mRNA, you can actually find the ones that are related to an inflammatory response, versus for PCR, it's not as useful. And that's why we're using RT-qPCR. Yeah, that's it. Any questions?
Any questions?
Hello. Hello, good evening, and this is Bharat. This research, you mentioned some medicine, for example, the prednisone and, the acetaminophen. Those are, I'm using now, I have some problem with my skin, and then I'm using those ointment, and then I take the prednisone as well, still taking some of them because I have something wrong with my liver. So I'm very interested, these things. And one thing is that, you are you trying to replace this medicine with the new technology?
Not really. We're just trying to like make better ones by adding to like dexamethasone specifically. Yeah.
Better one, and then you extract from the plant.
It should be, well, I think these are already like, they're not naturally occurring, but they're already like synthesized, and we already get like in lab we already have like the dexamethasone in a box, and then you kind of just like work from there. But I know so cortisone is naturally occurring. Yeah.
It is effective. Yeah, it's much more effective, and you only maybe identified the chemicals, identify the gene, and then you sequence it. Yeah. So is there any chances to clone it in E. coli or something to transfer in other plants? Do you think so?
I think that would be really cool to do. I haven't really thought about that, but I think, that's a good idea.
Because as a plant tissue culturist, if, future medicine, as far as I know, is not in the market, but now the scientists are saying that there's a medicinal horticulture, medicinal crop. Now they are thinking to make these any kind of drugs within the plant, within the fruit trees, so that, for example, if you have a problem of whatever the cancer or those things, and the chemical, if they are effective, we can clone it and then we can transfer it into the, for example, in the banana, and the banana plant will be the resistant to they have to use as a medicine, as a tablets in the future. So maybe I think this is the time to think about it as well, so that the future prospect of medicinal horticulture will be coming soon. Within 1 or 2 decades, the market will capture by the once you eat one banana, maybe you need not to take any vaccine for flu, because that might be cloned with the flu vaccine already. That is what I'm thinking, and then I'm trying to, in my lab, maybe we can clone. If you get the clone, we can transfer it into our crop. So maybe in the near future, you can think on it as well, I think. And your research, no one believed that it is done by the high school graduates. You are so smart. You are so nice.
Right.
Everything perfect and very clear. Congratulations.
Thank
Okay.
you.
You are the future scientist, I guarantee, because you have a very clear concept and then you are in the very good track. Is your opportunity, as I already mentioned, you are in the best place in Silicon Valley, and then this is the place utility. And then time, you are in the best time to involve in the organization to do this kind of research in the, before graduation from the high school. Congratulations for your whole
you.
team. Thank you.
If there are no more questions.
All right. Thank you. Good job. Good job.